So many epithelial cells transport ions and other small molecules from one side of the membrane to the other. So things can go in either direction depending on what function and what functions are required. So for example, epithelial cells lining the stomach secrete hydrochloric acid into the stomach lumen. And that's why your stomach is very, very acidic. The epithelial cells are secreting acid into the stomach lumen, making acidic. And when you get reflux, of course, you're refluxing up your esophagus that hydrochloric acid, which gives you that heartburn. The epithelial lining, lining the small intestine, transport products of digestion into the blood. So they're moving the other way from the GI tract into our body, into the blood. Another important concept for you to understand, which many of you will already, is that plasma membranes have an electrical potential difference. In other words, they have a voltage gradient across them. This is also called the membrane potential. This will be particularly relevant when you come to look at the nervous system. And the whole point about this is, here's your plasma membrane, here is your cytoplasm, so that's the inside of your cell, and here's your extracellular fluid outside the cell. And as you can see here, there are lots and lots of sodiums and potassiums outside the cell, so in your blood and in your extracellular fluid. And there are lots and lots of potassiums inside your cells, okay, in the cytoplasm. Okay, but overall, just because of differences in ion concentrations across those membranes, basically because of ion pumps, so sodium and potassium pumps, there is a net negative charge inside the cells relative to outside. Now, when you come to the nervous system, you'll go through this more. But it's just an important concept for you to think about or have in the back of your mind at this point. So, how do we transport things across the epithelial cells? Well, we need to transport things from the apical, that's the luminal side, so that's shown there, okay, to the basolateral side, which is shown here, so for example in the extracellular fluid, okay, and that's termed absorption or alternatively reabsorption if it's already been in once and it's being reabsorbed back in again. So that from the lumen to our blood or our extracellular fluid, so that includes our interstitial fluid and our plasma, that is called absorption or reabsorption depending on the tissue. Transport from this basolateral membrane to the epithelium, to the apical, the luminal side, that is called secretion. So when we secrete, an example would be hydrochloric acid, secreting acid from the basolateral membrane through the apical to the lumen and into our GI tract, or into our stomach in this case. So have a little look at this. Oh, I've just helped you, which is rather unfortunate. Have a look at this. So in each case, there are 3 little blanks in there. I just want you to tell me which one's absorption, which one's reabsorption, which one's secretion. Okay, press pause, have a read, and then I'll go through it. Okay, so uptakes of nutrient from the lumen of the GI tract, which is there, is termed, so they've gone from the apical to the basolateral, that is absorption. Transport of sodium chloride and water from the lumen of the renal tubules back towards the blood is termed reabsorption, because it's come in once, it's got on its way out, and it's coming back in again. And transport of hydrochloric acid into the stomach is called secretion. Okay, so if we're looking at epithelial transport, molecules need to cross these epithelial layers to reach that final destination. So we just talked about it. We talked about how hydrochloric acid does it. We talked about sodium chloride needs to come back from the kidney tubules, for example. And we can also think about it in terms of drugs, as well as nutrients and other molecules. We can think about it in terms of drugs. And drugs is a very important concept for you to understand. So, for example, oral drugs, so you take a drug orally, must cross the epithelial layers of the GI tract before entering the bloodstream. So you may take your paracetamol or your aspirin, for example, or your ibuprofen. You take that, comes into your GI tract. Of course, that's in tablet form. It has to dissolve out first, okay? So it has to break down. It has to dissolve and become soluble, okay? If it's still in a particulate form, it's not going to cross any membranes. So it needs to do that. So what it must do, it must cross these epithelial cell layers before crossing the basement membrane and the endothelial cells, those are the cells lining the blood vessels, before it gets into the blood itself. Of course, drugs can be taken by aerosol, topically, intramuscular, intravenous, but we're thinking orally right now. It's got to get across that epithelial cell layer. So how do drugs and other molecules, for that matter, how do they cross? So here's your cell membrane, okay, and within that cell membrane you have transporters, different types of transporters that help to transport molecules across a membrane. So you have diffusion, you have passive diffusion, and that's really driven by concentration gradients. So they go from a high concentration gradient to a low concentration gradient. For example, passive diffusion of gases are very permeable. So this lipid membrane is made of lipids. So if this is lipid soluble, it crosses really easily. If it's water soluble, it doesn't cross through this passive diffusion very readily because water can't dissolve through a lipid membrane. You then have facilitated diffusion. These basically also go down a concentration gradient from high to low gradient, but they go through a transporter, so they're facilitated, they're helped. Okay, and this might be water-soluble molecules or water-soluble drugs, okay, that can't cross by passive diffusion across a membrane because it's made of lipid. They pass with the aqueous environment, so water comes down this channel and your molecule, your drug, or whatever it might be, follows. Okay, so that's facilitated diffusion. And then you have active transport. So this uses another protein molecule that uses ATP, which is your energy form, to transport the molecule across the membrane. Now this might be against the concentration gradient in this case, but sometimes, with drugs, for example, it's still with a transport gradient, but this is the only way the drug can cross. It needs to use one of the cell's transporters that uses energy. I'd like you to look at this little table and just tell me, do each of these occur by passive diffusion? The answer is yes or no. So, yes or no to different degrees, obviously, but yes or no is what I want. So passive diffusion is high to low concentration across a lipid membrane. So gases, for example, CO2 and oxygen, yes, they do cross by passive diffusion. That's what happens in your alveoli, in your lungs. They pass. CO2, as it happens, is far more readily diffusible across a membrane than oxygen is. So sometimes in a variety of different lung disorders, you get different effects on carbon dioxide and oxygen transport across membranes or diffusion across membranes. Okay, just because CO2 is more readily diffuses across a membrane. Hydrophobic drugs, that means water hating, so lipid liking. So lipid liking drugs, do they cross by simple diffusion? Yes, they are lipid, so they cross the lipid membrane very easily. What about hydrophilic drugs? In other words, water liking drugs. They don't cross very easily by passive diffusion because they are water based, or they dissolve in water and they can't cross. They might use facilitated diffusion, for example, or they may come across by active diffusion. Active transport, rather. So, what are some of the factors that affect absorption across epithelial membranes? And in this, again, I'm just bringing drugs into this because drugs with medical students is one of the things that that people tend to find the hardest to understand. So I'm trying to get you to think about how drugs, in this case, but any molecule, okay, how its various factors affect its absorption across epithelial membranes. So on the left-hand side, okay, so these first two columns, this is the drug factor that affects absorption, okay, across a epithelial cell membrane. And so, for example, it's molecular size. So how big is it, big or small? And what will be the effect on the absorption rate? So all I want is increased rate or decreased rate. So for example, molecular size. If something is bigger, do you think it'll go across the membrane faster or slower? Well, of course, you're going to get a decreased rate. It's bigger, so it's clunkier. It's harder to get across that membrane. Okay, so what I'd like you to do, have a think about each one. So in other words, press pause, have a think about each one, then press play again. Okay, ionization. Okay, so ionization, basically the more ionized a drug is, the more water soluble it is, and therefore the more water soluble it is, generally less of it can cross a membrane because the membranes are hydrophobic. But what is ionization? What does it actually mean? A lot of you will know, but some may not. So think about table salt, sodium chloride. Okay, an ionized table salt is sodium ions, Na+, and chloride ions, Cl-. So when you put salt into solution, it can get ionized into sodium molecules or sodium ions and chloride ions. And so the more they separate apart, the more ionized they are, the more water soluble they are. So in other words, the greater the ionization, the slower the rate, because they're more water soluble, they can't cross a lipid membrane very easily. Lipid solubility. If you increase the lipid solubility of a drug, of course, it can cross a lipid membrane much easier. Structure's a hard one for you, okay, because it varies, because that depends on its solubility, how soluble it is in water and lipid, for example, its ionization state, its molecular weight, how big it is, its dissolution rate, that's just how fast it breaks down from tablet format, for example, and any transport mechanisms that might be in place. So that varies. And likewise, your dosage form, that also varies. So it might be a tablet, it might be IV, it might be intramuscular, it might be topical. Okay, so that again, that varies. Tablets, for example, dissolve at different rates. So there are simple little things. So now we're looking at the body factors. So what are the body factors that affect absorption? So surface area. So if the surface area increases, what will be the effect of absorption? Well, of course, that will increase as well. Okay, because you've increased the surface area, there's more capacity, more space for these molecules, drugs, for example, to cross over. The pH, the acidity or the alkalinity, that varies. So, for example, acidic drugs like paracetamol are less ionized in acidic media. So you take paracetamol, put it into an acidic media, it becomes less ionized. So, in other words, pH may cause some drugs to be less soluble. For example, like I said, acidic drugs are less ionized in acidic media and are therefore more lipid soluble. And ditto for basic drugs in basic media, examples being adrenaline, morphine, cocaine, for example. Vascularity, the amount of blood going past that tissue. So that increases. The more blood flowing past that tissue, the more the drug or the molecule can get in to those blood vessels because there are more of them. The number of cell membranes, that leads to a decreased rate. For example, the number of cell membranes, you have lots of different layers in your skin, and each drug or other molecule has got to get through one layer, then another, then another, then another. So you get a decreased absorption rate due to the distance it has to travel and the molecules it has to get through, for example. We're thinking about your GI tract, your food presence. Food in the GI tract takes up space, if you like, decreases the surface area for any drug that you might have taken, for example, because it's taking up that space, so you get a decrease in your rate. And gastrointestinal disease, the last one. So if you have gastrointestinal disease, remember when you're absorbing drugs or even your general nutrients that you're eating, there's a huge surface area in your GI tract. And if you have GI tract, you may have lots of that damaged and your surface area decreases, for example. So therefore, you may get a decrease in the rate of absorption or transport across a membrane. Okay, so we've really done some of the key things about the epithelium now and transport mechanisms. But there are some very important definitions that you need to know. So you need to know what diffusion is. We've kind of talked about it. You need to know what bulk flow is. We will talk about it. You need to know what osmosis is. We'll talk about that too. And really for later on, but we're going to touch on it now just because it's an important concept and it's very common. You need to know what edema is as well. Okay, so I'll just, again, press pause. Think about what is each of these. Give me a definition. Okay, so diffusion. We know what that was. That's the passive movement of molecules or particles from a region of higher concentration to a region of lower concentration. We did that earlier, so that's nice and easy. Bulk flow. Before I go into bulk flow and osmosis and edema, actually, I just want you to have a little look at this diagram on the right-hand side. Really, it's quite simple. Here's your blood vessel, that's the red, and inside there you've got your plasma. Remember, your blood vessels are lined by endothelial cells. And then outside that, so in there you've got your plasma, then you've got your cells and your tissues, and inside your cells you have intracellular fluid. And actually that's where most of the water in your body is. It's inside cells. But the fluid that bathes the cells, so in other words surrounds them and surrounds your tissues, that is called interstitial fluid. Now you'll understand from this, from a basic concept, when you eat food, the nutrients get into the plasma and they have to get to the cells. So what they do is they have to diffuse through capillaries out of the plasma, out of the blood, into the interstitial fluid, and from the interstitial fluid into the cells. And wastes go in the other opposite direction, so carbon dioxide and lactic acid and all other kinds of wastes diffuse from the cells into the interstitial fluid, then into the blood, where they get processed, for example, in the kidneys or the liver, for example, to get removed. Okay, so little concept for you to think about that many of you will already know. But what is bulk flow? Well, bulk flow is the movement of fluids from high to low pressure, also known as filtration. Okay, so for example, if you're thinking about your capillaries, that's where your exchange of nutrients goes on. So we'll call this a capillary for sake of argument. Say this right-hand side, blood is coming from the heart. It's now made its way to the capillaries where you can get some exchange. This is our capillary, and then this is where it's coming out as a vein. At this point, where it's coming from the arteries and the arterioles, we're at a higher pressure. So pressure, that bulk flow or that filtration pressure, forces fluid from the blood vessels into the interstitial fluid. Now it's a protein-free plasma that goes there. So your ions and your water, so your electrolytes, your sodiums, your potassiums, your glucoses and other things pass through into the interstitial fluid, and then they can go to the cells as required. But protein does not go through, so it's a protein-free plasma. But we're losing lots of fluid by doing that because you've got that bulk flow happening. So water needs to move back, or water, electrolytes and other stuff needs to walk back at the venous end of the capillaries. So basically fluid works there at the arterial end, goes towards the cells, towards the interstitial fluid, and at the venous end it comes back in again. So it maintains your blood volume, really, in essence. Okay, so that's bulk flow. The first flowing is bulk flow. You're forcing the fluid out. Osmosis is the movement of water across a semi-permeable membrane. For example, your cell membranes, your red blood cells membranes, for example, from a high water concentration to a low water concentration. So in other words, you've got loads and loads of water with very little solute, so other stuff in it, to a lower water concentration where there's water there, but there's also lots of other stuff in it, so the water concentration is lower. We will come back to that in a second. And then you have edema. Edema is accumulated or increased fluid in these interstitial fluids, in these interstitial fluid spaces. So you have increased fluid in there. That is edema. You also might know it as swelling. There are lots of different types of edema. There's inflammatory edema. So when you get inflammation, a sting, a bite or something like that, fluid builds up in your interstitial fluid, and that's inflammatory edema. Okay, so you've got fluid build up there. Okay, so like I said, do you know your definitions? So remembering what osmosis is, a very, very important concept to understand. This is movement of water across a semi-permeable membrane. So in other words, it's permeable to water, but not to some other stuff. From a high water concentration to a low water concentration. You can think of it the other way, from a low solute concentration to a higher solute concentration. So, in other words, water moves from a low sugar concentration to a higher sugar concentration when there's a semi-permeable membrane in its way. And this therefore makes the sugar concentration the same on both sides. Now you may still be a little bit confused, so what I want you to do is look at this diagram. Here we have a beaker. There we have a nice beaker. In it we have a semi-permeable membrane. This membrane is impermeable to solutes, so glucoses, sodiums, whatever those little green bits are. It doesn't really matter what they are. This membrane is impermeable to those, but it's freely permeable to water, which is what all this space is. That's all water, of course. Which way is the water going to move by osmosis? Which way is it going to move by osmosis? What it wants to do, it wants to make the concentration of the solute the same on both sides. So that's what it's going to attempt to do. Now, hopefully you said the water is going to move from left to right. So in other words, water is going to leave this side. Water is going to leave this side, move into that side. So the water level on this side might lower to there, okay, where my little arrow is. And the water layer on this side might go up to, better not go above the container, might go to there, okay. That's because the water has moved. And it does it until the concentration of the solute is the same on both sides. So that's osmosis, movement of water across a semi-permeable membrane. So we've talked about electrolytes a little bit. And what are electrolytes? Well, electrolytes really are ions, okay? And they're released when inorganic ions dissociate. And as the example I gave you was sodium chloride, dissociating into sodium ions and chloride ions. So this, again, very important concept in terms of the nervous system, okay, and the generation of nerve signals throughout the body, because they work on how many ions, an ion gradient across membranes. But also it's very important in terms of what happens within the rest of your body. If you have too much potassium, for example, too much potassium in your extracellular fluid, or too little potassium in your extracellular fluid, that can lead to arrhythmias and potentially heart attacks or myocardial heart attacks. Okay, so that's just something for you to bear in mind. So I just want you to look at this. You don't need to know the concentrations right now. We will expect you to know probably by the end of this year, because it's a very important concept, that you understand or know what a normal concentration of potassium is outside the cell and sodium outside the cell. It doesn't really matter what it is inside. You don't need to worry about that, because clinically you measure sodium and potassium a lot in your plasma samples and chloride to a lesser extent. Okay, so just tell me, concentrations of those three ions inside and outside the cell, I just want high or low in each case. Okay, potassium inside the cell, that is high. Sodium inside the cell, low. Chloride inside the cell, low. Now, what are the concentrations outside the cell? In other words, in your extracellular fluid. That includes your interstitial spaces and your plasma. Potassium, low. Oh, sorry. Important point. All of those things added up together, the overall, it's called osmolarity, but really it's like concentration. The overall osmolarity or concentration of all of the sodiums, the potassiums, the chlorides, the other inorganic ions, the proteins, the glucoses, everything that is in there adds up together to give an overall concentration or osmolarity of 290 milliosmoles per litre. Okay, you might see it written milliosmoles per kilogram. Don't worry too much about that, it's pedantics. So 290 milliosmoles per litre. Now, outside the cell, potassium, low. Sodium, high. Chloride, high. So what you're seeing is they're inverse with each other. Okay. Now, what is the concentration or the osmolarity in our extracellular fluid, presuming you're nice and healthy? Do you think it's 290? Do you think it's above 290? Or do you think it's 200? Or do you think it's lower than 290? In health and normally hydrated individual, it'll be the same, 290 milliosmoles per litre. And remember what that is. That's adding up all the potassiums, the sodiums, the chlorides, the proteins, the glucoses, and everything else. Everything adds up to 290 milliosmoles per litre outside the cell. Everything adds up to 290 milliosmoles per litre inside the cell, but they're different things, okay? So, or different amounts of things. So lots of potassium there, not a lot of potassium there. Loads of sodium and chloride there, not a lot of those two there. There's a lot of phosphate here, for example, that's a buffer. Not very much phosphate outside the cell. But overall, they add up to 290 milliosmoles per litre on both sides. That means the osmolarity or the concentration is the same on both sides, which means there is no osmolar gradient. In other words, there's no osmolarity difference, so there is no net water movement by osmosis because the solute concentration is the same on both sides of the membrane. This cell is a semi-permeable membrane that will allow water to go across it in either direction depending on the concentrations. Okay, so again, I'm going to get you to think about this again in a second now. So, capillary functions. Remember what the capillaries are there for. They allow the diffusion of nutrients from the blood to the cells and the waste from the cells to the blood for excretion. So here we've got our blood flow. Remember what happens? Blood flow, bulk flow comes through, blood pressure forces fluid into the interstitial fluid without the proteins. Proteins stay in the blood. Comes this way and it's now into the interstitial fluid and it can now exchange nutrients with the cells. Okay, and at the venous end of the capillary, fluid moves back down into the blood to maintain that blood volume. Okay, and we won't talk about the mechanisms for that for now. And so remembering what we got, I've just said this. We get the filtration, the bulk flow of protein-free plasma out of the capillary into the interstitial fluid, and the reabsorption of water and solutes into the capillary. Okay, but the capillary wall is highly permeable, okay, to water and solutes, but not to protein. Very important. So all the solutes and all the water can pass through there nice and easy. Okay, it's basically got a little, it's like a little sieve, but it's not big enough to allow those sieves, those holes aren't big enough to allow proteins. So what happens as the fluid leaves here, goes lots of fluid, lots of fluid, lots of fluid, starts to bathe the cells, the blood volume goes down, but the protein concentration goes up because the volume has gone down, but the protein amount of protein is the same. So therefore at the venous end there's a huge amount of protein, relatively speaking. So fluid now moves from the interstitial fluid where there's very little protein into the blood where there's lots of protein down its osmotic gradient from low protein concentration to high protein concentration, or high water concentration to low water concentration. That's the basis of fluid movement through capillaries. Okay. And something you also need to know, how much fluid have we actually got? How much water, rather, have we got in our body? So in an average 70 kilogram male, because many years ago when the original textbooks came out, they used 70 kilograms as the average weight of a male. There is 42 litres of water. 28 litres of that is in your intracellular fluid. That means inside your cells. 11 litres is in your interstitial fluid, the fluid bathing your cells. 3 litres is in your plasma. I want you to think for a second. I want you to tell me what would be the effect of eating a high salt diet on fluid movements between compartments. So basically, I want you to think, you've just eaten a whole load of salt. Okay, I'll just say, for sake of argument, I've given you 2 teaspoons of salt, and you've eaten 2 teaspoons of salt straight into your stomach, and you haven't got any other food in there, okay, to mess around with us. So you've eaten that salt. Where does it get absorbed into first? What does it do to that compartment in terms of osmolarity? And therefore, what does it do in terms of osmosis, water movement from one compartment to another? So I want you to think about that. Press pause again. Have a good think. And then when you think you've got the answer, come back and listen and see if you have. Okay, so hopefully you've had a good think about that. So what does the high salt diet do? So if you've had a load of salt in your diet, that salt, that sodium chloride, comes into your blood. That's where it's absorbed from your GI tract into your blood. So the osmolarity of your blood goes up. Okay, it's normally at 290 milliosmoles per litre. Let's say you've eaten enough salt and your blood osmolarity has hit 300 milliosmoles per litre. So now you've got 300 milliosmoles per litre in your blood, but only 290 in your interstitial fluid and 290 in your cells. So we've now got an osmotic gradient between our blood and our interstitial fluid. So water's going to move one way or the other. Which way is it going to move? So water would leave the interstitial fluid and move into the blood to try and dilute the blood. Okay, so now the interstitial fluid has nicely diluted our blood a little bit back, but we've also taken water away from our interstitial fluid. So our interstitial fluid now, say for sake of argument, is 295 milliosmoles per litre. Okay, the same as the blood. Okay, I'm not giving you accurate numbers. Doesn't matter what the numbers are. It's a principle. So now that's at 295, but our cell was at 290. So we've now got an osmotic gradient between the cell and the interstitial fluid. So what happens? Water's going to move. Water's going to move from the cell into the interstitial fluid to make the concentration the same on both sides. So the cell shrinks. Okay, so that's what happens if you have a high salt diet. Basically, plasma osmolarity goes up, and then you get this rapid flux of movement out of your cells, out of your interstitial fluid, into your blood to dilute your blood down. It's not happening in these discrete steps that I just tried to explain it to you. It's in flux. It's all occurring very, very rapidly and together. But the whole point is that at the end of it, your plasma osmolarity will be higher than it should be. Okay, it'll be 295, 296, whatever it is, it doesn't matter. It's no longer 290. It's a little bit too high. And because it's too high, you might find that you're starting to feel thirsty. So even though your blood volume has gone up because you've stolen water from your interstitial fluid, you've stolen water from your cells, your blood volume has gone up, so you shouldn't be dehydrated, but you'll be there with your mouth going, oh, I'm thirsty, doing those sounds where you've had a high salt diet. You'll be wanting to get water, and you'll want to be drinking water to try and dilute the blood even further. And that, as you start to drink, that increases your blood volume, which increases your blood pressure. Hence why a high salt diet is bad for you. It increases your blood volume, therefore increasing your blood pressure. And over many millennia, our bodies are not particularly good at excreting sodium from our bodies. They're very good at retaining it and getting it, but they're not so good at excreting it. And some of us are poor at excreting the excess sodium that we eat in our diet. Hence the increase in blood pressure. Okay, that's it.